Hello and welcome back to This Week in the Clinic. So happy to be back with you today. We've got a super interesting topic, something that has, I think, gone or come into the consciousness of many people's minds over the past few years, especially those who are in the health field. And we're talking about genetic testing and the role it plays in functional medicine. For those of you who might be joining me for the first time, my name is Josh Gitalis. I'm a clinical nutritionist and a functional medicine practitioner, and I have a practice here in Toronto, Ontario, which I own and operate, and I also work with people all over the world through Skype and phone. I also run a functional nutrition certification program, and we are beginning again in September with a whole new group of students, so I'm super excited about that. But today, we're going to get into genetic testing. It's a super interesting field. It's one that's expanding hugely in the research field. We're learning way more about it. And as such, a lot of people are becoming more and more interested in it. So that's what I'm talking about today. We're going to talk about a little bit about the history of genetic testing and how we got into this whole world of looking at our genes. What exactly genetic testing is. Some of you may have not even heard of genetic testing before. We're going to look at the role of genetic testing in functional nutrition and functional medicine, how we can actually use it to make clinical changes and to make measurable results in people's health. Why genetic testing can be misleading. So there are a lot of misconceptions around it. Let's look at what those are and what the actual pros and cons are of genetic testing. How to use genetic testing for powerful results. And finally, how to get what was once a $3 billion test for just a few $100s. So let's begin. So, starting out in the 80s, I believe it was, the U.S. government got really interested, and I think the world got really interested in mapping the human genome. So there was an approval for the Human Genome Project, and it began in 1990, and they estimated it would take about 15 years to map the whole human genome. They beat their goal by about 2 years and did it in about 13 years. And they had a budget of about $3 billion. Okay? So a pretty expensive, project, sequestering researchers from all over the world to help out in this effort. It ended up costing about $2.7 billion, so they were able to save a few dollars here and there. But the price of this has actually come down substantially. What was once obviously a completely, inaccessible test to the average person has now become accessible to each and every person, which is pretty amazing. So this was $2.7 billion to map the human genome, back in the 90s. They eventually did it to completion in 2003, so it took them about 13 years. But today, we can actually get the same test for about $250, which is really cool. But because it has become so accessible, a lot of people have lost, or maybe don't even know, the utility of using this test to actually make changes in their health. And that's what I want to get into. But before I get into that, I just want to talk a little bit about what the difference is between genotype and phenotype. Okay? So we're all born with a certain set of genes, half from our mother, half from our father. We actually have about 25,000 genes. And anyone know, who's joining me live, how many genes a piece of rice has? Humans have about 25,000 genes, but how much does a piece of rice have? Anyone have the answer? Well, the answer is 100,000 genes. So it's quite interesting, The first complete genome that was mapped was the human genome, but then we started to look at all sorts of other plants and animals, and what we found out is that actually, mushrooms are quite close to the human genes, and of course, primates are very close to the human genes as well, but that a lot of plants even, like rice in this example, actually have more genes than humans. So it wasn't necessarily the more you have, the more advanced you have, which is kind of interesting. So our genotype is our book of life, right? We have our genes in the center of every cell, it's the nucleus, and it gives the information for the whole cell so that it can operate, so that it can do what it needs to do on a daily basis, make all the proteins, process, toxins, repair different cells, tell when cells need to replicate, when cells need to die. There's a lot of information in there that directs our whole body to work the way it should work and to keep things in what we call homeostasis. And the old paradigm was that we thought the genes were our destiny. Whatever genes you got from your parents, that was going to determine your health throughout your lifetime. So if my father got lung cancer and my father's father got lung cancer and his father's father got lung cancer, well, guess what? I'm going to get lung cancer. But now we know that that old paradigm has been completely disproven. And now we have a new paradigm telling us that, the genes aren't that important. It's that how we express those genes is where the importance lies. And that's called the phenotypic expression of the gene. And I'll give you an example. In about 50 years ago, the chance of getting breast cancer, if you had the breast cancer gene, was about 24%. Today, it's about 80%, okay? So over 50 years, the human gene hasn't changed at all. It actually takes thousands and thousands and thousands of years for that human gene to change, but our environment, our food, what we bathe that gene with has changed drastically. And that's what influences our genes to be expressed in certain ways. And that's a term that we've given to it is epigenetics. Epi meaning above, genetics meaning genes. So we're really looking at what's called epigenetics here today. And I'll give you another example of how genes are expressed. If you think about a butterfly, right? How does a butterfly start out as their life? They start out as a caterpillar, right? And at some point that caterpillar completely encloses itself in a cocoon, dissolves, restructures itself into a butterfly, and emerges as a completely different organism. Now, the genes are the exact same, but there have been certain things that have taken place that change the way that gene is expressed. Another really good example. Another one I'll give you. So we've got different types of bees in a beehive. We've got the queen bee, and we've got the worker bees, and there's hundreds and hundreds of worker bees, but there's only one queen bee. They decide when they need a new queen bee at some point in the hive's life, and when they do, they help develop that queen bee into the queen of the hive. And this queen bee is 3 times larger than a worker bee, and it lives about 4 to 5 years, whereas a worker bee will live only a few months. Now, believe it or not, but they just randomly choose which egg is going to be that queen bee. And the genes are absolutely identical to the other bees in the hive. But they feed that queen bee a very special food. Does anyone know what that very special food is? No one else in the hive gets it. The other bees eat honey. The other bees will eat some bee pollen. But this one bee, the queen bee, gets one single food that no other bee in the hive gets for the duration of her whole life. And that is royal jelly. With the royal jelly, she becomes extremely fertile. She grows 3 times larger and lives up to 4 to 5 years, which is way more than the other bees in the hive. So this is an epigenetic influence on that bee's genes. Same genes, but they're being expressed in a completely different way. And really, that queen bee's job is to just procreate with the other bees, just to get it on with lots of bees in the hive. She makes about 2,000 eggs per day at the height of her fertility. So That's a big job, so she needs some good food for that, and that's what the royal jelly provides her. We've also seen studies done where we take mice, completely identical mice, and we deprive them of certain nutrients. They're called methylators. There's a study done where they deprive them of vitamin B12 and folic acid. And what they found was that the mice that didn't get the methylators became obese and actually had different color hair on their body. Very, very interesting. So exact same genes, different expression. So how does that translate to humans? I think that's where the power lies. So I'm going to use a few examples to help illustrate this point and how we use it in clinical practice. And we'll look at an example as well. So I think coffee is a really great example as to understanding genetic uniqueness and how we all express in a different way. So there's certain companies out there today that believe every human and their children and their dog and their hamster should be consuming coffee. Now, being as a nutritionist, I've seen hundreds of clients, and I believe that each person is biochemically different, and not one thing is good for everyone. And we see through genetic testing and through what our genes tell us that this is actually, in fact, true. So a study was done on Costa Rican men where they looked at caffeine consumption and heart disease risk. And what they found was, the average Costa Rican man would consume about 4 cups of coffee a day. And some would get heart disease and some wouldn't. And they wanted to know if there was any correlation with caffeine consumption and if that had an influence on their heart disease risk. And what they found was that the individuals who would have 4 cups of coffee per day and were fast metabolizers of caffeine had a decreased risk of getting heart disease. The ones who consumed 4 cups a day but got heart disease, were slow metabolizers of caffeine, had an increased risk of getting heart disease. This goes back to our genes, and we can actually test to see if you're slow or fast metabolizer in your genes. How cool is that? So I'm actually a slow metabolizer. My wife is a fast metabolizer. We both checked our genes. So it's fascinating. If we're living in the same household, and oftentimes people in the same household have similar habits, and we both consumed caffeine or coffee on a regular basis, my wife would be getting benefits from that based on this study, cardiovascular benefits, whereas because I'm a slow metabolizer, I would actually be getting a detrimental effect. And that's possibly due to some of the polyphenols in the coffee. They're not outweighing the benefit of maybe having to detoxify the caffeine or other constituents in that coffee. This is and the enzyme responsible for this is cytochrome P450 1A2. Now, if you don't know what the hell that means, don't worry about it. It's not that important. But this is the gene that's responsible, and we can test it to see if it moves fast or slow. So if I got that information, I find out I'm a slow metabolizer, that tells me that, caffeine might not be the best thing for me if optimal health is what I'm looking for. Or I want to consume caffeine in some serious moderation, right? And that's how we can use this information. Now, just another piece to it, the way you feel isn't always a good indicator. So I used to be a coffee drinker many years ago, and when you consume any type of drug, your body actually creates tolerance to it. It increases detoxification pathways to process that drug a lot quicker so that it doesn't harm you. It's a protective mechanism. So I was able to have, 1 to 2 cups of coffee, no problem. Sometimes it wouldn't even stimulate me that much, and that's because I ramped up my detox pathways. And that's my phenotypic expression of that gene. Even though I'm a slow metabolizer, I've sped it up. So sometimes when people have been exposed to a certain drug or a certain chemical for a period of time, they're not necessarily able to get a good reading whether they're a slow or fast metabolizer. So that's one great example using coffee and caffeine. And there's a company which can map your whole genome. Remember I told you it used to be $3 billion and now it's about $250. Their company name is 23andMe. And one of the things that they give you in the reports is whether you're a slow or fast metabolizer of caffeine. Pretty interesting stuff. So I'm going to give you another example on how we use it quite effectively in our clinical practice. So we use genetic testing a lot with individuals who might have hormone imbalances. So for example, women who are worried about possibly estrogen-dependent cancers, maybe they've had some breast issues, maybe they have a history of breast cancer, and they're worried, or perhaps there have been some suspicious things happening like fibrocystic breasts, or maybe some benign lumps in their breasts, or maybe they've had an abnormal pap smear result. Different things that point to possible hormonal imbalances, we can measure those hormones to see where the balance is. So let's talk about estrogen for a moment. If I go to a conventional medical doctor and I ask them to test my estrogen, they're going to test one estrogen. It's called estradiol. Estradiol is one piece of the big hormone picture. And there's actually many different estrogens. There's three main types of estrogens that our body makes and creates. One's estradiol, one's estrone, and one is estriol. Estriol has a much more protective effect than the other two estrogens. It's anti-proliferative, whereas the estradiol and the estrone can be proliferative if they're in high levels. So, with functional lab testing, we can actually dig a little deeper and see which hormones are in what amounts and if they're in balance. But that's not where the story ends. When we make these hormones in the body, we still have to detoxify them, process them, and get them out. And we do this through a two-phase process called liver detoxification. There's a phase 1 and a phase 2. I'm not going to go into details there. If you want to get into details, join me in my detoxification course, where we go through it in great detail. But our body converts these estrogens into less harmful or different types of estrogen so that we can poop them out or pee them out. Now, in order to do that, we need enzymes. Enzymes are the workers of the body. They change things, they manipulate things, they break things down and build things up. And we can actually test genetically to see if we have any mutations in these enzyme genes. So I'm going to take a quick break here to explain what a lot of these genetic tests are looking at. We get one gene from our mother and one gene from our father. And they have, in scientific language, we use 4 letters for them, A, G, T, C. Okay? That doesn't matter, but basically 4 letters make up our whole genetic sequence in different combinations. So one from our mother, one from our father. And we have sort of the normal type of gene. So if that's normal, then that gene is just working at a regular rate. When there's one mutation from one parent, then that gene can work either a little bit too slow or a little bit too fast. When there's mutations from both genes, both parents, that gene can work very, very fast or very, very slow. The effect is magnified. These mutations are called single nucleotide polymorphisms. And if you take the first letter of each of those words, they're called SNPs, is the short form for that. And these SNPs really help determine why we're genetically different, right? Why some people process caffeine quickly, like my wife I was mentioning, Megan. I see she's actually joined us on this live stream. Whereas I am a slow metabolizer of caffeine. I don't process it as fast. And that's because I have certain SNPs that slow down those detoxification pathways. So going back to the hormones and how I can use this information clinically. So one thing we do is we will test to see how many of those estrogens and what levels they are in the body, right? And that's the phenotypic expression. But what a lot of people don't know and may not appreciate is that there's really 4 really important genes that help to sequence for the enzymes that detoxify those estrogens. Does that make sense? So there's 4 genes. So we can see, just by testing the hormones, what the phenotypic expression is. But if we want to know what that person's genetically susceptible to and how to maybe support those pathways, we can look at the genes as well. And that's how we combine the best of both worlds, right? It's not just about looking at the genes and saying, oh my God, I've got a mutation here, I've got a mutation there. It's going to be the end of the world. We also have to look to see how they're being expressed and if that's a problem. One of the big genes, that's one of the best research genes, called MTHFR, and it has to do with folic acid metabolism and the methylation pathways. And when people find out that they have this mutation, sometimes they freak out. I've even had people come to see me just based on that mutation and say, hey, I've got this mutation, what could you do about it? But it's not just what gene we have, but it's how it's expressed. And what are things that help that gene express properly? So even though it's moving slow, if we're doing the right stuff, we can support its activity and that person may not suffer any ill results. So exercise helps it, sunlight helps it, lots of green leafy vegetables help it, lots of good water. So all these lifestyle things that we know are positive interventions actually help the gene do what it does. So I'm going to give a couple other examples, using myself as an example. So I've tested my genes, and another thing that I found out is I have an increased risk of getting Alzheimer's and Parkinson's disease. Well, again, I could look at those results and say, oh no, I'm on the path to neurodegenerative disease. That's what my genes say. I can't escape it. I might as well just go out and party every day. No, it doesn't really work like that. I actually find this information incredibly empowering. So in the general population, about 7.2% of the population are going to be diagnosed with Alzheimer's disease. My risk was about 10.4% in relation to the population. So I had a 1.46 times increased risk of getting Alzheimer's. For Parkinson's, about 1.6% is at risk will be diagnosed with Parkinson's in the population. My risk was 2.51 times higher than the general population. So we know that with Alzheimer's, it's actually being labeled in a lot of research papers as type 3 diabetes. Why? Because when you have years and years of blood sugar regulation disorder, that can really affect neurological health and actually cause brain shrinkage. Dr. Amen, who's a world-renowned neurologist, says, big body, little brain. Basically, the more fat you accumulate, the smaller your brain gets. It's called brain shrinkage. And actually, we have direct correlations with blood sugar and hemoglobin A1C. As those go up, brain shrinkage goes up as well. Quite interesting stuff. So I'm just going to check in here, see some comments, and then I'm going to come back and give you the other part of the story for the Alzheimer's and Parkinson's. So I see Megan. Megan has written a joke that she's just sitting around chugging coffee at 38 weeks pregnant, and she's just kidding. But she is a fast metabolizer. That's my wife, for those of you who don't know her. And as I spoke about before with the research, she would be able to process it a lot quicker. Please feel free to post any questions as I go through here. And then also, Megan, we tested her genes, and her risk for Crohn's and colitis was 10 times higher than the general population. Now that's really interesting, because Megan actually was diagnosed with Crohn's. Now, if she looked at those results many, many years ago, would she have looked at them and said, I'm definitely going to get Crohn's? Maybe not. It might have empowered her to do everything she could to avoid that path, because we know that there's certain risk factors associated with Crohn's disease and inflammatory bowel disease. like the consumption of sucrose. People with inflammatory bowel disease consume 2 times the amount of sucrose per year. We know that formula can increase risk of inflammatory bowel disease. The standard American diet, which is high in inflammatory fats, can increase inflammatory bowel disease. So that was actually the path that Megan was on back in the day before she got diagnosed with Crohn's. She got diagnosed, did a total lifestyle revamp. All of this great information is in her book, Undiet, and she also has a cookbook with a whole bunch of recipes, but the whole story's in here, and she ended up learning how to heal the body and went into nutrition, and the rest is history. So it doesn't necessarily mean what's in your genes is your destiny, because we saw an expression of it, and then it got completely reversed. Now, in conventional medicine, they think that if you get a specific disease, oftentimes a lot of these autoimmune diseases, that you have it for life. And you go to a lot of these websites and they say, we're fundraising for a cure. We're searching for a cure. We're fighting for a cure. And the cure that they're often looking for is in the pharmaceutical world, a drug that cures an individual. But that's just not possible. The only way we cure the body is when we go through healing, right? Now, when it comes to pathogens, that's a bit of a different story. We can actually cure people by killing certain pathogens when it's a pathogen-related disease. But when we're talking about chronic and degenerative diseases, it's a completely different story. Okay? So we can change the way those genes are expressed. Just going back to some comments here and questions. Someone says, Kel says, I know I have the MTHFR gene mutation, Alzheimer's gene, genetic heart conditions. So with an unhappy face, and that's exactly what I'm talking about today, is that we don't have to have unhappy faces. As I mentioned, I have increased risk of Alzheimer's and Parkinson's. But because of that, and because I know that, I'm going to take measures to really prevent it, right? I know that those are maybe my genetic weaknesses. So I'm going to try to manage my blood sugar over a lifetime. We know that certain heavy metals can really affect neurological health, and some of the genes associated with increased risk of Alzheimer's tell us that people with those genes have a decreased ability to clear mercury from their brain. So if I had silver amalgam fillings and I found this out, I'd get them out right away. Now that I know this, way before that has happened, I try to avoid mercury as much as possible, eating the right fish if I'm going to eat seafood, and staying away from mercury products, right? I'm not going to put amalgams in my mouth because I know I'm more susceptible to the damage there. And we know that meditation helps to prevent neurodegenerative diseases. So I'm going to take part in meditation on a regular basis. We know that caloric restriction helps with preventing neurodegenerative diseases. So I'm going to try not to overeat, which is sometimes a challenge. We know that exercise helps to prevent neurodegeneration. We know that the fatty acid omega-3 DHA helps to prevent neurodegeneration. So I'm going to do all these practices because I have this knowledge that I'm more genetically susceptible. And that's how we can really change how our genes are expressed and use this genetic information to our advantage, right? It's not there to freak us out or to scare us. It's there to empower us. And I talked about the breast cancer gene before, and I've spoken to a lot of women who say, I don't want to know if I have the breast cancer gene. If I have it, that's just going to freak me out. But I would think completely opposite. If I knew, if I was a female, men get breast cancer too, so I'm not discounting all men as well. But if I knew I had the breast cancer gene, I would do everything I could to make sure my hormones were balanced, to decrease my risk factors. We know that smoking, obesity, not consuming enough vegetables, wearing a bra more than 12 hours every single day, taking the birth control pill, all these things play into breast cancer risk. So I'm going to start to control these things as much as possible. I'm going to do some preventative hormone testing because, again, no conventional doctor is doing hormone testing to check for prevention for things like breast cancer and hormone-related cancers. So I'm going to do that on a regular basis. I'm going to check my genes to see if I need to support any genetic pathways to make sure that I'm detoxifying hormones appropriately. There's so much we can do, and it's all about empowerment. It's not about being freaked out. And if the genetic information does freak you out, work with a functional medicine practitioner or functional nutritionist to figure out how you can best support those pathways. Karen says, I'm excited to see my results soon. The neurological connection fascinates me. Yeah, it fascinates me too, and I'm sure you will be enlightened. Maria says, what would be the difference between saliva test and this one? I'm not quite sure what you're referring to when you say this one. The saliva test is actually how they gather your DNA. That's what 23andMe uses. You basically spit in a tube, send it in, and they get some, skin cells in there, and they're able to map your human genome. Lynn says, would the average person be able to read the results of 23andMe, or would they need to see someone like you? That's a great question, Lynn. And it's really like, back in the day when you had the microscopes in chemistry class or biology class, and you'd look through the microscope and it'd be like 40 times and then 400 times and then 4,000 times and just get more and more magnified. Well, these genetic results, it's a rabbit hole, and you could go as far deep down that rabbit hole as you want. 23andMe makes a really user-friendly report on their website. So some of the information I was talking about today regarding caffeine, metabolism, Alzheimer's, Parkinson's in relation to my specific situation were just things that 23andMe provided. And even on their website, you can go deeper and deeper into the research and go further. I use a couple third-party pieces of software to get a little bit more specific to look at genes associated with hormone detoxification, as I was talking before. So there's a great website, Genetic Genie, and you can get a free report from them on methylation and on detoxification. So those can be helpful if you know how to read them. Again, that's more advanced, but you can slowly chip through it and look up each genetic mutation to find out what it means. And another third party that does a very comprehensive report, it's about 18 pages, might be a little bit TMI for some people, is Sterling's app. And if you just type in Sterling's app in Google, you'll find it. You plug in your raw data and you get an 18-page report. So that's a little bit much for people, but if you know how to read it, it helps. So that is my discussion on genes for today. Do I have any more questions? Just please put them in if you do. If you like what I'm saying, give me a thumbs up or a heart. I always appreciate that. And if you know that someone else would benefit from this information, send it off to them. Tag them. Share it on your page. Let them know that this is what we're chatting about today. And it's all about, when we're talking about genes, how we can combine both the genetic genotype information, what's in the book of life, with our phenotypic expression and how we're actually expressing those genes. So I started out this discussion by saying the old paradigm said if my father got lung cancer and if my grandfather got lung cancer, my great-grandfather got lung cancer, I'm going to get lung cancer. Well, that's not necessarily the truth. It's about how those genes are expressed. And, if they all smoked, they're turning on all these cancer genes in their lungs and turning off all the protective genes. If I stop smoking, I've done one lifestyle modification that's going to greatly decrease my chance of getting lung cancer. So that's what it's all about. We want to take this information, know how to interpret it so we don't get confused and freaked out, apply it to our lives to make positive changes, and really maximize our genetic potential. And there's 7 billion people on the planet, and there's 7 billion different biochemistries, and therefore we all need different foods, different lifestyle interventions, and even different relationships. Lynn says, great info. I was scared to get this info, but after your presentation, I'm going for it. Yes! that's what it's all about. I'm happy we got you thinking a little bit differently about the genetic information. So I want to thank you all so much for joining me for this week in the clinic. I'm Josh Gitalis, and the best way to get started is to get started. Take care and have a fantastic